Hi everyone, welcome to your Chapter 12 video. If you are watching this in the fall of 2020, I want to remind you that you will have a quiz this Friday. It is going to cover chapters 12 and 13, okay? So this week, our topic of conversation is going to be all about cell cycles and cell division, okay? What that means is that we're going to cover some topics that you have some degree of familiarity with. In particular, we're going to be covering mitosis in chapter 12, and we're going to be covering meiosis, in chapter 13. So use these two chapters that you may have some level of familiarity about. Use these as your opportunity to really study hard and get some credit on this next quiz. Hopefully you'll find this material to be somewhat familiar to you. Now, in thinking about where we're going in chapter 12 and what we're going to be talking about, we're going to be focusing on the cell cycle. So you can really think of this as sort of like the life cycle of a cell, almost like you studied the life cycle of a butterfly or the life cycle of a frog. We're going to study the life cycle of a cell. And I want us to think about the life cycle of a cell, particularly for eukaryotic organisms, as occurring in two phases, okay? We're going to talk about cells just having a phase where they're just functioning as the cell. They're just living their best cell life. If it's a liver cell, then it's just doing all the detoxification that liver cells do. If it's a brain cell, it's doing all of the neurological things that brain cells do. And this period of cell functioning, we're going to talk about as being the portion of the cell cycle called interphase. Then we're going to spend a lot of time talking about what happens when the cell is ready to make a copy of itself. What happens when the cell is ready to multiply? And that is going to happen via two processes that I want you to know about: mitosis and something called cytokinesis. Okay? So let's draw ourselves a roadmap for where we're headed in this chapter. First of all, we're going to talk, as I said, about cell cycle and cell division. We'll put cell division. We're going to talk about it in eukaryotes first. And that's where we're going to talk about things like interphase and mitosis. Then we're going to talk just briefly about cell division and how it occurs in prokaryotes. And that's going to happen via a process called binary fission. And then the very last thing that I want to talk about is how the cell controls cell division. So we'll put control of division. And actually, I want to focus a bit on what happens when the cell loses control of when it divides. Because when that happens, you end up with the disease that you know as cancer. Okay, so cancer and the formation of tumors is actually the result of error in control of cell division. So it's actually an error in mitosis, okay? So that's where we're headed with chapter 12 today. So I want to first focus in on the cell cycle in eukaryotes. And to help you understand the cell cycle, I want to give you a mnemonic device, okay? I want you to think of this nonsense word, ip-mat, okay? It's not a real word, right? But it's the initials for some parts of the cell cycle. And I think if you learn this word ip-mat, it's going to help you understand what's going on. So what do each of these initials stand for? First of all, the I stands for interphase. Okay, interphase, if you were taught about it in high school or in middle school biology, you probably learned that it was a period of just normal cell functioning. We'll put cell growth and, we'll put cell growth and functioning, okay? And by cell growth, I mean an individual cell getting bigger. So normal cell growth and function. Okay. After interphase, you've probably been taught that cells can replicate or divide using a process called mitosis. Okay, and mitosis can be divided up into 4 different parts or 4 different phases, okay? And that's what each of these other initials stand for. So the first phase is called prophase. The second is called metaphase. The third is anaphase. And then lastly, we have telophase, okay? Now, you were probably taught that mitosis is cell division, but what I'm going to teach you today is that mitosis is actually the division of chromosomes. So what we're going to be doing, chromosomes. So what we're going to be doing in mitosis is we're actually going to have 2 copies of the DNA in our cells. And the goal of mitosis is to put 1 copy in each of 2 new cells, okay? So that's what mitosis is going to do. Mitosis is going to result in, take 2 copies and result in 1 copy in 2 new cells. Now, there's a process that is actually related to mitosis called cytokinesis. And this word is kind of a self-defining word. The word root cyto means cell. So anytime you see cyto as part of a word, it's referring to something cellular. The word kinesis means movement. Okay, so what happens in cytokinesis is once you have a copy of your DNA here and a copy of your DNA here, cytokinesis splits the cell membrane to make 2 cells. So we'll put splits the cell membrane. Okay, so when we think about cell division is actually a combination of mitosis plus cytokinesis. I'm put cyto K. These 2 things together equal cell division. Okay, now let's break this down into some detail, all right? So recall that I told you, you could think of the cell cycle of eukaryotes as being sort of like looking at the life cycle of a butterfly or the life cycle of a tadpole. So here, this is a diagram of the life cycle of a cell, okay? And I'm gonna start here at the top. This M part right here stands for mitosis. okay? And then you can see we also have cytokinesis. So let's start here where we have a cell that's divided and we have a brand new, fresh baby cell. Immediately after a division, okay? So we have a baby cell. That baby cell is going to spend some time growing, so going from being a small cell to getting to be a little bit bigger. And it's also just going to spend some time functioning, doing whatever its job is as a cell. Okay, so this period of growth and functioning, this first period here, as you can see, that is called the G1 phase. G1 stands for gap 1, and I'll tell you about how it got that name in a little while. But just know that what's happening during the gap 1 phase is that the newly divided cell is just growing and functioning, kind of living its best cell life. Now, how long will a cell stay in G1 phase or gap phase here? Well, that really depends on the type of cell that you're dealing with. So for example, if we were talking about a brain cell, what happens is that as you develop as a little baby, a little embryo, and then a little baby, you're making neurological tissue. But there comes a point in your development where you have made all the brain cells that you're going to make in your lifetime. Those cells become stuck in this G1 phase. They never divide again, and they never regenerate. And so that's why a traumatic brain injury is such a big deal, because if you were to like hit your head and damage your brain cells, those cells don't divide, and therefore your brain tissue cannot regenerate. Now let's contrast that with, say, a cell that lines your intestine. You know, every day you eat, hopefully you're eating 2 to 3 times a day. And as you do, your muscles of your stomach and intestine are like contracting to force that food through your digestive tract. And that food, as it's running through, it's causing the cells that line your digestive tract to peel off, to slough off. So you're constantly having your intestinal cells divide so they can replace the ones that you lose. So if you're an intestinal cell, you don't spend very much time in this gap phase, this G1 phase at all, because you're in it for a minute and then you divide, and then you're in it for a minute and then you divide, okay? In contrast, if you're a brain cell, you might spend hopefully 80 or 90 years in gap phase, because hopefully you'll live a nice long life and your brain cells will stay with you the whole time. All right, so let's just assume that we're talking about a cell type that does divide, something like an intestinal cell. So after a period of time where we're growing and functioning, then there's going to be some sort of a signal that says, okay, it's time to start preparing to divide. And the first thing that's going to happen to prepare us for division is we're going to enter something called the S phase. The S stands for the word synthesis. Now, you guys know that synthesis means to make something. If you're synthesizing something, you're making it. In this case, what you're synthesizing is DNA. So what's really happening during S phase is we are copying chromosomes. Let's think about what that really means. That means that during this phase here, if you were to peer into the nucleus of a cell, you would find all of the DNA necessary for that cell to function. You would find one copy of the instruction book for how to make cells in your body. After you finish going through S phase, you're going to have 2 copies of all of your chromosomes, okay? 2 copies of your DNA when you get to the end of this S cycle here, this S phase here, all right? It's these 2 copies that are going to get divided up during mitosis. But before we get to mitosis, we have one more phase of the cell cycle that we have to go through. And that phase is called G2, or the gap 2 phase. And what's happening here is that the cell is really ramping up for division. So it's making all of the enzymes that it would need for cell division. It might be doing things like copying organelles. And I'll just put preparing to divide. Oops, to divide. Okay. So we've gone through a phase of growth and functioning. After the cell has grown and functioned for a while, then it's going to copy all of its DNA so that at the end of the S phase, we have 2 copies of each chromosome. Then what's going to happen is the cell's going to enter a second gap phase, okay? And that second gap phase is going to serve the purpose of preparing the cell to divide. Once we've done all of those things, we're going to enter this mitosis phase here, okay? So here we are, mitosis. And I told you guys before that mitosis is actually dividing up the chromosomes . Cytokinesis is splitting the cell in half, okay? So at the end of this M phase, what you will get is you will get 2 cells. And here's the important bit. These 2 cells will be exact copies of one another. Because remember, you made 2 copies of your DNA here, right? And all you're doing in mitosis is you're separating the copies out. So you've got 1 copy here and 1 copy here. Cytokinesis is going to split the cell up, and we're going to go through how all this happens. And so you're going to end up with 2 identical cells as a result of mitosis and cytokinesis, okay? Now, really quickly, I do want to tell you a little bit about how we ended up with this terminology of gap 1 and gap 2. So I'm sure that this isn't the first time that you've heard something about mitosis. I'm virtually positive that in your high school biology classes, you've seen images of mitosis. Sorry, I had to drink some water. Okay, so if you kind of envision in your mind's eye what mitosis looks like, you can probably envision some like darkly stained chromosomes inside a cell. So mitosis was actually really easy for early scientists to observe through a microscope, okay? So I'm gonna put observe, and I'm gonna put scope for microscope. So once we had microscopes and cellular stains, so we could like stain the cell membrane and we could stain the chromosomes, we could actually see this occurring. So for the longest time, we defined a cell's life as being just interphase, which I'm going to come back to in a second. And we thought of interphase as this period of dormancy, and then we had mitosis, which is where we thought all the action was, right? But because that's what we could see. We didn't know any more than that, okay? I just misspelled interphase. It should be an A right there. But once we got a little more sophisticated about our science, some folks ran experiments. And one of the things that they did, I'm going to erase in here, was they fed some cells radioactive phosphate groups, okay? So these were radioactive. And then they followed where the radioactive phosphate groups went. Now remember, if you've got a molecule of DNA, let's do a little review here. Okay, and then you have a base here, and you have a base here. Here's another phosphate. Okay, so if you remember what DNA looks like, it's a sugar, phosphate backbone. So if you feed a cell radioactive phosphate, I'm going to get rid of that real quick. If you feed a cell radioactive phosphate, it can get incorporated into DNA. So they watched when this radioactive phosphate became incorporated, and they realized it was kind of right here at this phase of the cell's life cycle. But they realized that there was a period before the radioactive phosphate was incorporated and a period after the radioactive phosphate was incorporated. And so they just named these periods gap 1 and gap 2, and that's where that terminology comes from. Now let's get back to this idea of interphase. So I told you that, scientists for a long time, and I'm going to write it over here because I'm a lefty. So interphase. So for a long time, people might have described interphase to you as kind of a period of dormancy where the cell's not doing much. But it turns out that interphase has a lot going on. Interphase, if we were to diagram it out, is actually all of this starting at G1, going all the way through the S phase and up through G2. All of that is interphase. G1, S, and G2 is all interphase. Okay, so if we go back up here, where I said interphase is where normal cell growth and functioning is occurring, that's true. Other things that occur during that phase would be DNA synthesis, and preparation for dividing. And that is because interphase is made up of G1, where you have cell growth and cell functioning, S, where you have DNA synthesis, and G2, where you have preparation for cell division. Okay? So now that we know something about interphase, going back here, all of this here is interphase. Now we know something about interphase. I want to zero in on this section here where the cell is going to divide, okay? And I want us to talk about mitosis, and I want us to talk about cytokinesis. Let's start with mitosis. And I told you that mitosis really has to do with the division of chromosomes. So let's take a look at what's happening to chromosomes during the cell cycle. So hopefully you can see here that this is the same cell cycle I just taught you about, only now it's linear instead of being circular. So here's G1, and this is G1 right here. Here's S, and here's S right here. And here's G2, and there's G2 right there. And then we have our division bit here, and that's the M phase, and that is this right here. Okay, let's look at what's going on. So in G1, we have, I'm just gonna erase all this so we can see. In G1, we have the newly formed cell, newly formed here, kind of undergoing growth and cellular function. And in this example, we're going to say that we're dealing with a cell that has 1, 2, 3, 4 chromosomes. And you can see that they're living their happy chromosome life. Then we undergo the S phase, which you remember stands for DNA synthesis. And I told you that what's really happening here is we are copying the chromosomes, because you guys know that the DNA in your cells actually occurs in a chromosome form, right? So we are copying chromosomes, and what you should be able to see is that by the time you get to the G2 phase, can you make out that where we had one chromosome here, it's kind of doubled up, isn't it? And here's another one, and it's kind of doubled up. The same here, we got double, double, and double, double, okay? Then we enter the M phase. Now I want to point out something to you. Your book has drawn this specifically the way it has for a reason. Here we see that these chromosomes are long and skinny. And here you can see that there's long and skinny, but they're copied. So there's 2 of each of them. Here, I want you to notice that the chromosomes are like little X's and they're short and bulky. And that's actually on purpose. We're going to talk about that in just a second. Okay, so what happens is after G2, we have these 2 copies of each chromosome. The M phase is actually going to take these chromosomal copies and it's going to give 1 copy to one cell and then the other copy to the other. And it's going to do that for each of these guys, okay? We're going to walk through that process. Now, since mitosis involves the separation of chromosomes, let's learn a little bit about the structure of chromosomes, okay? So, when we think about the DNA in your cells, you often probably, in your mind's eye, you're probably just thinking about a double helix, right? So you're just thinking about something that looks kind of like this, boop, 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 right? But DNA is really long and it's really skinny. And there's a ton of it in your nuclei. Like in a human cell, if I were to take all of the DNA out of just one of the nuclei of one of your cells, and I was to stretch it from one end to the other, we would end up with a 6-foot length of DNA, okay? That is really, really long. And so the problem is that if we just shove that very long DNA into your nucleus, it might get tangled up. Think about a ball of yarn. If you didn't have it wound up into a ball and you just kind of shoved it into a basket or something, that yarn would become very easily tangled up. So in your nucleus, your DNA is organized, okay? So we're gonna say that your DNA is organized. And it's organized so it doesn't become tangled up. And the way that it's organized is just like you would wind thread around a spool to keep it organized, we're going to treat the DNA like a thread, and we're going to wind it around some proteins, okay? So this is in my little diagram here. This is DNA. And then this here is what I'm calling a spool, just like if you had a spool of thread. But these spools are actually proteins, and these proteins have a name. These proteins are called histones. Okay, so when you have a combination of DNA and histones together, like you do here, that is called chromatin. DNA plus histones are called chromatin. So during interphase, which you guys know is G1, S, and G2, if you look inside the nucleus of a cell, so here's a nucleus of a cell right here, can you see that this DNA is kind of loosey-goosey? It looks like spaghetti. But if you were to actually zoom in on it, you would find that it actually occurs as chromatin, which is DNA wound around a spool to keep it organized, okay? But if you were to look at it under the microscope, what you would see is something that looks kind of like spaghetti. Now, what happens at the beginning of mitosis, and I think I spelled beginning wrong, but at the beginning of mitosis, what happens is these histones start to stack on top of each other. And so at the beginning of mitosis, the DNA, the chromatin condenses. It condenses to form what you think of as a traditional chromosome. So the DNA condenses into chromosomes, okay? So basically what happens is this spool will stack on top of that one, and that one will stack on top of that one, and that will continue until you end up with what you've probably seen before as a very traditional chromosome shape, which is sort of an an X shape. So this traditional X shape is probably what you are more familiar with, but it's really only what we see during mitosis, okay? Now let's look a little bit about, look a little bit at the structure of a chromosome. So if we were to actually dissect it, you would see that what it is a bunch of chromatin that's all linked together, and it's because those histones have stacked up against one another, okay? Basically what we have here is a structure that is joined at the center at a region called the centromere. Let's write it out. Now, when you have this X shape, what you're really looking at is two different copies of the DNA. One copy here, and then a second copy here, okay? So I like to think of this almost like two pieces of paper that are duplicates of one another, okay? So this is one copy, this is the other copy. And the centromere is where these two pieces of paper are stapled together, all right? Another piece of terminology that you need to know is that each of these individual bits here, each individual copy, is called a chromatid. And we'll put that equals an individual copy. And because these chromatids are copies of one another, these two chromatids together are called sister chromatids. Okay? So make sure that you understand this anatomy. Make sure that you understand the centromere is what's holding these two copies together. Think of it as the staple that stapled two copies together. Each of these copies, this one and this one, they're each called a chromatid. Together, because they are copies of one another, we call them sister chromatids. And remember, you're only going to find chromosomes in this formation during cell division, okay? You're only going to see them like this during cell division, not during interphase. So when I say cell division, I mean mitosis. And then later on, we're going to talk about meiosis in chapter 13. Those are the only two times that you're going to see the chromosomes in this formation. During interphase, you're going to see the chromosomes look more like this spaghetti here, but what they really should look like is kind of like beads on a string where the DNA is wrapped around like thread on a spool. Okay? All right. Now that we understand how chromosomes look, let's talk about how we divide them up. Okay? So let's begin at the beginning of mitosis. Let's see here, I'm gonna get my notes. I have a very specific way I want to lay this out for you. All right. So the first stage of mitosis, so let's see here. First stage of mitosis is prophase. What does that look like? We're going to do some drawing. Now, let me first draw a cell for you before it enters prophase. This is a really simple cell. We're just going to draw the cell membrane here. So this is the membrane. And we're just going to draw the nucleus here. And I'm going to leave a few gaps because remember, the nucleus has some pores, some nuclear pores. But in general, that's our nucleus. And inside, our DNA is going to look something like this. Okay? Because before we enter cell division, before we get to mitosis, this is not condensed. So what's going to happen during prophase? I want us to look for 3 events that are going to happen during prophase, okay? The first event that I want you to notice is that we're going to have the nuclear envelope breakdown. Okay, so let's think about this cell here. This nuclear envelope, if we're trying to separate out the chromosomes, if we're trying to separate our copies of our DNA, this nuclear envelope is almost like a fence that's fencing in this DNA. So one of the first things that we have to do during prophase, here's our cell membrane. One of the first things that we have to do is we got to break down that nuclear envelope, okay? So I'm just going to draw it like this so you can see that it's in the process of breaking down. The next thing that we're going to see happen is we're going to see that the chromosomes condense. In other words, we're going to have all this chromatin, and I'll label that as chromatin. We're going to have all those histones stack on top of one another to form the more characteristic X shape. And I'm going to use some color coding here. So I'm going to do, let's see, I'm going to make a couple of large chromosomes like that so we can keep track of them. And then I'm going to make a couple of small ones, like we'll use blue. And just have them be small. So you can see that what's happened is we've gone from this big noodly spaghetti mess to these distinct X-shaped bodies, okay? And in my example, there are 4 chromosomes, okay? I've got 2 big ones in orange and 2 little ones in blue, okay? So I have 4 total chromosomes in this example that I'm drawing for you. Where we need to go is we want to have 4 chromosomes in our starting cell, and we want to have 4 chromosomes in each of the cells when we're done. Now, be careful, because if I ask you how many chromosomes, let me go back to black, how many chromosomes there are in the cell, some students want to tell me 8. They want to go 1, 2, 3, 4, 5, 6, 7, 8. Well, that's not correct. That student would be correct that there are 8 copies of the DNA. There's 2 copies of this, 2 copies of this, 2 of this, and 2 of this. But these copies are stapled together, right? So here's where it's stapled together, here we're stapled together, here we're stapled together, and here we're stapled together. So we only have 4 total chromosomes. The next thing that's going to happen during prophase is we're going to see the formation of something called the spindle apparatus. Now, believe it or not, we've actually talked about this before, apparatus. Okay. This is made of centrioles, and it's made of microtubules. So we've talked about microtubules before in chapter 7 as part of the cytoskeleton. So what's going to happen is you'll have these little cylinders, and they're kind of across from one another, and it's what makes microtubules. And these microtubules are going to start to kind of move out from the spindle apparatus, okay? And there's two of them in animal cells. There's two spindle apparatus. These are actually called microtubule organizing centers. And they're going to begin to form during prophase, okay? They're going to begin to form, and they're actually going to begin to try to attach, oops, hold on, to some of these chromosomes. Give me just a second. I've lost my place. Okay. So at the end of prophase, what's happened is the nuclear envelope has broken down. The chromosomes have condensed, and the spindle apparatus has formed, okay? So that's these microtubules here. Now, where are we going next? Well, the next place we're going to go is the second phase of mitosis, and that's called metaphase. In metaphase, what's going to happen is we're going to have a cell where the nuclear envelope is totally gone, okay? Where our chromosomes have lined up down the middle of the cell. So I'm going to draw the two orange ones, and then let's draw the two blue ones that were littler. Okay. So in metaphase, the chromosomes first, that's the first thing that's going to happen, is the chromosomes are going to migrate to the middle. So chromes migrate to the middle. Okay. The next thing that I want you to notice that happens during metaphase is that the spindle apparatus moves to opposite poles of the cell. So here's our centrioles. These are called centrioles. Centrioles make microtubules, so we're going to send those out. So these are all microtubules. So centrioles plus microtubules together, this is called the spindle apparatus. And what happens is the spindles move to opposite poles. And most importantly, the microtubules attach to centromeres. So here, what's going on is this microtubule is attaching to the centromere here, and this one's attaching here. This microtubule is attaching here and attaching here, etc., etc. Because hopefully you can see where we're going to go. What's going to happen is these microtubules are going to begin to retract in that direction and in that direction. And when they do, they're going to pull this copy here apart from this copy here. So these chromosomes are going to split down the middle, okay? That's where we're headed. So that's what's going to happen in the next phase, which is phase 3 of mitosis, called anaphase. In anaphase, the microtubules retract. And the centromeres, remember the centromeres are where it is stapling the chromosomes together. The centromeres split. Okay, so let's draw that out. Here's our cell. Okay. Here's our little centrioles. And here's our microtubules, because we've got 4 of them. And what has happened at this point is we split those chromosomes down the middle, and as these things start pulling in opposite directions, the little tails kind of flow out behind them, okay? So let's see here, boop. And just in case you're having trouble visualizing this, here is, I'm going to draw kind of an enlarged version of one of our blue chromosomes. And it had a big old centromere right here, right? What we had was some microtubules attached to this side here and microtubules attached to this side here. So when this one started pulling in that direction and that one started pulling in that direction, it caused this thing to split down the middle. And then as it pulled in that direction through the water, these little tails, they kind of drag behind. Okay, so that's why I've drawn it this way. The way that I remember anaphase, if metaphase is when the chromosomes migrate to the middle, so everything begins with the letter M, metaphase, migrate, middle. Here's how I remember anaphase. Do you see how these look like little triangles? It looks like a little triangle. And if you turn a triangle right side up and you draw a little bar in it, you have a letter A. So to me, this looks like a sideways letter A, sideways letter A. That reminds me that this cell is an anaphase, okay? All right, we've got one more phase of mitosis that I need us to go through. Hold on, I'm gonna add a slide here. Or am I? Am I gonna add a slide? No, I'm sorry, I'm not. All right, let's go back to anaphase here. And I wanted to talk a little bit about these microtubules retracting. Like, how do these microtubules pull in each direction, okay? Well, researchers wanted to know something about how these microtubules pull in each direction, so they performed an experiment. And what they did in this experiment, let me change colors again, use orange. What they did in this experiment was they labeled the microtubules with a fluorescent dye so that they would glow. And you can see that in this example, these yellow microtubules are attached here, 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 and here. Then what they did is they used a laser to darken this area here, okay? And that laser made that area black. And they watched to see what happened to the remaining glowing fragments, okay? Did they get longer? Did they get shorter, okay? So for example, did this region here get longer or shorter, or did this region here get longer or shorter? And what they found was, here's our little region of darkness. This region right up here actually got shorter. And that was the first clue that led us to understand what's happening to the microtubules during mitosis. So check it out. I'm gonna go back for a second. What we're gonna hone in on is this area here where the microtubule is attaching to the centromere. So right here, this is where the microtubule is attaching to the centromere. So this area would have been the centromere right there, okay? So the centromere is actually bound to a protein complex called the kinetochore. Okay, so here is where your centromere would be, and then this weird looking thing here is just called the kinetochore. And the only reason I bring it up is because I wanted you to know that there's like a little ring here. And what happens is, as what causes this to pull this direction is this ring causes the microtubule to fray, okay? So the ring causes the microtubule to fray. And basically, as it frays the microtubule, it pulls the ring in that direction, okay? And that causes the chromosome to then pull in that direction. So right here, to pull the chromosome, the microtubule disassembles, causing the microtubules to shrink, is what that should say. All right, so just to kind of go back where we were, I just wanted to talk to you a little bit about how these microtubules shorten, and they shorten because starting at this end, they literally disassemble and move backwards in that direction, okay? All right, one more phase of mitosis that I want to talk about is the last phase. It's called telophase. By the time we get to telophase, what's happened, I'm going to draw the cell kind of like an egg shape now. Because what's happened at this point is that the nuclear envelope is going to start to reform. So I'm going to kind of start drawing that in. The nuclear envelope is going to start to reform. And at this point, you see here how we have an orange, a blue, and a blue? All right, let's do that here. We're going to have an orange, a blue, and a blue. But what's going to happen as you go through telophase is this is condensed, and as you move through telophase, it's going to relax back into chromatin. So you'll have some spaghetti here that's orange, and some spaghetti here that's orange, and you'll have some spaghetti here that's blue, and some spaghetti here that's blue. So basically, at the end of this process, you still have 4 chromosomes, 1, 2, 3, 4. But what'll happen is as you move through this process, these things will relax. So I've drawn it right at the beginning of telophase and toward the end of telophase, okay? So now what we have are 2 identical nuclei. Okay, but we still don't have 2 cells. And for us to get 2 cells, what's going to need to happen is we're going to need to split the cell membrane down the middle. And that happens in a separate process called cytokinesis. Actually, it's one word, sorry. I don't know why I did it as 2. Cytokinesis, okay? And so what happens in cytokinesis is, remember talking in our chapter 7 video about a cytoskeleton element called actin, okay? So actin gets laid down across this middle boundary, and actin partners with a motor protein called myosin. So what happens is the myosin is going to move in this direction, and it's going to move in this direction. And as it does, it's going to cause the cell membrane to increasingly pinch in on itself until eventually you have 2 separate cells, and we already had our 2 identical nuclei. And let's draw our DNA. Here's our orange, and here's our blue. So the result of mitosis plus cytokinesis is 2 exact copies. Okay, this is an exact copy of this. This nucleus here has 1, 2, 3, 4 chromosomes. And if we go back to the very beginning when we started this, we had 1, 2, 3, 4 chromosomes. All we did was separate them down the middle to make 2 copies, okay? Question that you need to ask yourself is, review, how did you get these 2 copies in the first place? And I'll give you a hint, go back and review what happens during interphase. All right, let's look at an overview of this process using the figures from your book. Okay, so this is just a series of figures from your book that are doing the exact same thing that we just drew. Here we have an interphase cell, okay? It doesn't really tell us which part of interphase we're in. Let's see here. Oh, wait, let's see here. After chromosome, oh, yes, it does. After chromosome replication, each chromosome is composed of 2 sister chromatids. So if we're in interphase here, and we've already had chromosome replication, then I know we're in G2. And if that doesn't make sense to you, you need to go back and review interphase. So what we have here is a cell with a nuclear envelope, and you can see that all of, oops, what's happened here? Here it is. You can see that all of the DNA in there, it looks like spaghetti, so right now it's just chromatin. Okay, as we move from G2 into mitosis, what's going to happen is we're going to see the spindle apparatus form. We're going to see the nuclear envelope is going to start to break down, and we're going to see the chromosomes here start to condense, okay? So you can see that the nuclear envelope is breaking down. You can see that the microtubules, the spindle apparatus is present. Now, your book breaks prophase down into 2 phases, prophase and prometaphase. Just know that I lumped these 2 together into prophase, okay? Once we have condensation of our chromosomes, we're going to end up with a situation where we have our chromosomes have migrated to the middle, that's metaphase. Here is our microtubules, okay? And they have now attached to these centromeres here, okay? Moving on. The microtubules start to disassemble, and as they do, they pull the chromosomes apart with 1 copy going that direction and 1 copy going that direction. That's anaphase. In telophase, we're going to have the nuclear envelope reform. The chromosomes are beginning to relax, as you can see here. Then we undergo cytokinesis, where actin and myosin start pinching this thing in. Eventually, the cytoplasm becomes divided, and we end up with 2 identical copies. Okay, and hopefully this all makes sense to you, but on the off chance it doesn't, I'm going to change my share here. And we are going to watch a brief video demonstrating how the cell cycle works.
Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis or nuclear division, and cytokinesis or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into 3 discrete phases called G1, S, and G2. During the G1, or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing 2 copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has 4 stages: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed and the microtubules attach to each sister chromatid. Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. In telophase, the 2 groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, the chromosomes uncoil and the spindle disappears. The division of the cytoplasm and organelles is called cytokinesis, or the C phase. The result of mitosis and cytokinesis is the formation of 2 genetically identical cells. To summarize the entire cycle, G1, S, and G2 phases occur during interphase while the cell is growing and preparing to divide. Mitosis, or the division of the nucleus, consists of 4 phases: prophase, metaphase, anaphase, and telophase. And in these phases, duplicate copies of the chromosomes are split equally. Finally, the cell's cytoplasm divides during cytokinesis and results in the formation of 2 genetically identical daughter cells.
Okay, so
Most eukaryotes.
Hopefully that all makes good sense to you. I'm going to go ahead and stop this video here. I have just a little bit more to tell you about from Chapter 12, and I'll put that in a Chapter 12 Part 2 video. Talk to you soon. Bye-bye.